Yeah, awesome. Hi, everyone online and no one here. Um, my name is Z. I just finished my rotation with Claudia, Dr. Kleinman, and I worked a little bit on perturbed fish. If anyone of you heard of that project, I'll be presenting at the 3MT later today. So today our talk will be by is Dr. Selin Jessa? Dr. Selin Jessa. Who's currently working at the Wu Tsai Neurosciences Institute she is an interdisciplinary postdoctoral scholar and a CIHR Banting postdoc scholar in the William Greenleaf and Anshul Kundaje labs. Sorry if I mispronounced that, at Stanford University. She actually completed her PhD here in our program, the QLS at McGill, and also completed her bachelor's in the Joint Honors Comp Sci Biology program at McGill as well. And among the many fellowships, scholarships, and awards, there are two notable highlights. The Governor General's Gold Medal, which is awarded to the most outstanding graduating doctoral student in natural sciences at McGill, and also the Gordon A. McLachlan Prize, awarded to an outstanding graduate within the biological and health sciences. And today's talk, she will be discussing dissecting gene regulation syntax using glass box machine learning. Yep.
Thank you.
Thank you.
Awesome. Thank you, Z, for the introduction, and thank you, Alex and Celia and everyone for having me back at QLS. So yeah, I finished up my PhD in Dr. Claudia Kleinman's lab in April 2023, and then I moved to California to start my postdoc in September of 2023, and so this is what I've been working on for the last year and a half or so. And yeah, this is a very collaborative project. I'm co-supervised by Dr. Will Greenleaf and Dr. Anshul Kundaji, and we've worked on this project with Kyle Farr's team at Illumina AI. And I had three fantastic co-leads on this project, Betty, Sam, and Yan Ting. So the major question that has been motivating a lot of my postdoc work is trying to understand how a single genome gives rise to hundreds of cell types in the adult human body. Right, so most of our somatic cells have a near identical genome, and yet we have specialized cells in our eyes, our brain, our muscles, our skin, and so on. So how does a single genome give rise to all of this diversity? So the short answer is that genes are very precisely regulated. They're turned on and off at the right place and time during development. And the organization of DNA is what in part enables this regulation, right? So our DNA is wrapped around protein octamers called nucleosomes. This substance is called chromatin, and chromatin can be packed very tightly, or it can be more loose and accessible. And this level of compaction or chromatin accessibility can control whether genes are available for transcription or not. I'll be talking about this concept or phenomena of chromatin accessibility quite a lot. And just to get on the same page about some numbers, so we know that it's cell type specific expression of genes that lead to cell type specialization, but only about 1% of our DNA, of our 3 billion base pairs of the human genome, actually codes for proteins and is exonic DNA. But outside protein coding regions, we also have functional elements with a code, the cis-regulatory code. And so we're interested in specific functional regions of non-coding DNA. We call these regulatory elements. And typically when they're close to the genes at the 5' end, we call them promoter regions. And acting at these regulatory elements, we have transcription factors, which will bind DNA in a sequence-specific manner. They'll recruit co-activators and transcriptional machinery to activate gene expression programs. And these transcription factors will often bind a small set of sites, which can be summarized by a motif, which basically represents the collection of sequences preferred by a certain transcription factor. And we can remind ourselves, like, what does it mean for a transcription factor to have sequence specificity? And we can remember the structure of DNA as the double helix, right, where we have these rungs on a ladder represented, given by the nucleotide base pairs. And each base pair exposes a certain set of chemical groups, hydrogen bond donors and acceptors, and also methyl groups to either the major or the minor group, depending on which side we're looking. And these base pairs and chemical groups also affect DNA shape. And so together, the chemical surface formed by both the base pairs of DNA and also DNA shape itself are read out by transcription factors, and some of these chemical surfaces will be more favorable for binding of certain transcription factors. And so because of the diversity of binding domains on transcription factors and different protein structures, as well as the distinct chemical surfaces presented by DNA given by specific sequences, we have all these variety of sequence preferences among transcription factor families, with just a few examples shown here. One other element of the cis regulatory code that we're very interested in is that combinatorial transcription factor binding or synergy of binding can also contribute to cell type specific activity. And we can think of this as a way for the genome to achieve even greater specificity, for example, by requiring two proteins to be expressed and bound for activation of a certain gene, right? Like a logical AND gate. And importantly, these transcription factor binding sites could be arranged within regulatory elements with many different properties, so different spacing, orientation, order, composition, and so on. And so we can think of this like a syntax or a grammar of transcription factor binding sites that's embedded in DNA and might affect its function. And you're very familiar with the concept of syntax from natural language, right? And so the sentence, the dog chased the cat, is not the same as the cat chased the dog, even though they have the exact same collection of words. In this case, the meaning of the sentence is fully dependent on the order of words within that sentence. So we know that transcription factors are expressed in many cases in a cell type specific manner. They recognize specific DNA sequences. The syntax of binding sites within DNA can affect synergy and combinatorial binding, although it's not fully understood how the syntax mediates synergy. And so ideally, if we wanted to map transcription factors and their combinations in all the relevant cell types, we would measure the position of every transcription factor in every cell type. But one challenge is that binding of a large diversity, profiling binding of a large diversity of transcription factors in many human developing cell types is right now technically infeasible, often because we don't have the right antibodies for all these transcription factors, or because we need too much cellular material to run an experiment that would allow for profiling binding, or because those cell types themselves are inaccessible. So one thing that is tractable to do at scale is to profile chromatin accessibility using the next generation sequencing technique, ATAC-seq. And so the way this technique works is we use an enzyme, TN5 transposase, which will cleave DNA wherever it's accessible to that enzyme. And when transcription factors bind, they're including their binding sites from cleavage, but inducing local accessibility where other things can't bind. And so in the ATAC-seq experiment, we can sequence all the fragments that are cut by TN5 transposase. We can sequence them, and then we can count the number of cut sites given by the starts and ends of these fragments at every position in the genome to basically get these insertion profiles. And because of how transcription factors protect DNA from cleavage by the ATAC-seq enzyme, these ATAC-seq insertion profiles actually reflect both quantitative and qualitative features of transcription factor binding, such as the number and the position of transcription factors, right? So in this case, the red transcription factor is bound here, and you see this sort of footprint in the ATAC-seq profile, but also other properties like positioning of these transcription factors with respect to nucleosomes. And so we can use these ATAC-seq profiles to basically learn information about what transcription factors are doing on DNA. And I forgot to say at the beginning, I'm happy to take questions throughout, so please just get my attention if you have a question. And so, yeah, just to recap, we can basically measure chromatin accessibility at scale, including in single cells, to learn about the landscape of regulation in any given cell type, including the landscape of transcription factors. Yeah, there's a question from Cesar, I think.
Yes, hello, Selin. Can you hear me?
Yep.
All right, perfect. So yeah, my question is one of, I guess, probing the data a little bit. So the existence of this compositional grammar is assumed, right? But beyond these finding experiments that track with where we expect to find these elements, how or which litmus test can we run empirically, probing the data, to see if these syntax elements are actually present in the data that we're using to model, and then the volumes and quantities that they require to be picked up by the model as a significant contribution, let's say?
Yeah, it's a good question. So for the second question, what are some litmus tests we can use from the data to define that? I'll come back to that in the later part of my talk, where we're trying to define methods basically to pick out these elements of syntax in order to learn how they affect accessibility. But to the first question, the presence of these syntax elements is assumed. On the one hand, yeah, it seems logical to assume that such organization of binding sites is present. But actually, there are plenty of experiments to show that this syntax is relevant in many cases. And so there are experiments, for example, that will show that you can have certain affinities of binding sites that are not perfectly optimal, not the most strongest binding site. And in a mutant animal or like a mutant fly, for example, when you improve the affinity of that binding site, you suddenly get ectopic expression of that gene. That's one example. You have examples of transcription factors that will only bind DNA when they're in complex and can find their sites at a specific arrangement. So yeah, these are conceptually some of the theoretical elements of syntax, but there's lots of work to suggest that these syntax rules are operating, and the challenge is really how to systematically analyze them. Does that answer your question?
Yes, partly.
Okay, cool. I'll come back to the
for the
second one.
part, yeah.
Yeah, I'll come back to the second one later on.
Perfect, thank you.
Yeah, thanks for the question. Okay, so we're measuring ATAC-seq to learn about transcription factor binding, and how we've used that in our team is basically to use this technique to learn about transcription factors and their effects on accessibility and development. And so well before I got to the lab, the Greenleaf lab together with Illumina's AI team had been starting to generate this atlas of single-cell gene expression and chromatin accessibility in human to fetal development, where they've profiled about 800,000 single cells from 12 different organs in fetal development in the second trimester, and those organs are shown here. So I won't talk too much about this atlas, but I'll just give a very quick overview of this data set to set up what I'll talk about later. So we performed a standard single cell analysis, we annotated cell types, and in the end we obtained 203 clusters from among these organs. And so to ask about the overall relatedness between cell types across organs, we did a hierarchical clustering by correlating expression profiles, average expression profiles of each cluster. So in this tree, every leaf represents one cell type. You can see the annotation of organ on the top and the age on the bottom. And we get both very tissue-specific cell types like hepatocytes and cardiomyocytes, but also subtrees of cell types that are present across many organs like neural cells or immune cells. Okay, so how can we use this data to systematically learn the effects of transcription factors on chromatin accessibility? So I mentioned that we think that this information is within the quantitative and qualitative ATAC-seq signal. And so to extract that information, we're using a machine learning strategy. Just gonna go over here. We're using a machine learning strategy. And so the way this works is we train a deep learning model, a convolutional neural network, to use local DNA sequence, about 2,000 base pairs, to predict chromatin accessibility in the pseudobulk ATAC-seq profile for one cell type in the central 1,000 base pairs of a given region. This is done for one cell type, so each model learns to predict the cell context-specific profile accessibility from DNA sequence alone. And the models are tasked with predicting two outputs, both the total accessibility counts, like fragment or read counts in a region in a peak, as well as the profile of accessibility, essentially as a probability distribution over that central 1,000 base pairs. And these models are called ChromeBPNet, and this strategy has really been pioneered by Anshul Kundaji's lab. So what's the purpose of tasking the model with predicting both of these outputs? One way to think about it is that instead of just having to predict the probability of a peak or the quantitative level of a peak, we're adding further constraints to the model and adding further constraints to what the model must learn to ensure basically that the prediction problem is sufficiently determined. And so the second question might be like, what is the point of these models? We already have this accessibility data. Why do we need to predict it? And our hypothesis is that if these models can successfully learn to perform this task, to predict chromatin accessibility from sequence, then they've learned something about the sequence rules that influence accessibility in this specific cell context. And we think that this works because these models reflect a causal phenomenon, like a causally consistent flow of information, where DNA sequence can affect chromatin accessibility, but not really the other way around. So how are we going to figure out what these models learned? For this, we use model interpretation strategies, specifically here, a technique called deep lift or SHAP. And intuitively, the way this works is by comparing the output predictions for a given genomic region to basically the output that you would get if you shuffled the sequences in that region several times, you take the average, and so you get, in a way, a reference value. We take the difference between the observed output prediction or the output prediction for the observed sequence with the reference value, and we can back propagate that through the network, weighted by basically the learned weights on these nodes until we got all the way back to the input layer, essentially assigning a score or a blame to each nucleotide, representing how much it contributed to the predicted accessibility in that region. And so, yeah, I'm happy to answer some more questions about how this works, but I'll save the details for another time. But essentially what I think is like a really nice paradigm about this method is that whereas typically machine learning models can be thought of as black box models, where we don't know what they're doing or what they're learning, and that can impede kind of the adoption of these models. In this case, we can think of this like a glass box model, where we can use such a technique to sort of peer inside the model and understand what it learned about accessibility. And what sequences it thought were important, and then ultimately do downstream analysis on top of these sequences. So we've trained and interpreted this type of Chrome BPNet model for every single cell type, all 203 cell types in our data set. And so we have these tracks now of these contribution scores across all peak regions in every single cell type. And so we can scan these contribution scores to pull out the contiguous sequences of high contributions, or basically the sequences of predicted importance. We can cluster them both within and across cell types. And ultimately, what these clustered patterns represent are very consistent with known transcription factor binding motifs that are experimentally derived, despite the fact that the transcription factors don't really know, the models don't know what a transcription factor is or what their preferred binding sites are. So we can annotate these motifs, we can use them to basically go back to our contribution scores and annotate the motif patterns that each of these high contribution regions match. And so now we basically have these in-genome sets or in-genome tracks of predictive instances which match the motif patterns based on sequence, but importantly, they also are predicted to influence either positively or negatively local chromatin accessibility in that peak. So how well does this work? These are examples for a couple regions where we have a couple genomic regions in one cell type where we have the observed chromatin accessibility from ataxic in blue and the predicted profile in red. And you can see that by and large, although the predictions aren't perfect, they capture the major signals, the major peaks and troughs in the ataxic data. And because of this model interpretation approach combined with our annotation strategy, we now have these annotation tracks for every peak region in every cell type, where we basically can see the contribution according to the models of each nucleotide to accessibility, as well as these automatically annotated predictive motif instances. And so I haven't added these manually. We have these tracks that we can now analyze. And we can think of basically these high contribution regions as inferred transcription factor binding sites. So we can also quantify the performance of these models. Like I said, these models have two outputs: a counts task and a profile task. And so the way we assess performance on the counts task is by looking at correlation between the predicted counts and the observed counts across peak regions. So this is kind of that scatter plot for one cell type where every point is one region. And here's the distribution of that correlation, of the Pearson correlation across all of our cell types. And so what's interesting here is that we can get a pretty high correlation of about 0.8 an R-square of about 0.65 using just local DNA sequence alone. And so for the folks watching who study gene regulation, you might ask, what about all these other levels of the cis regulatory code, like 3D genome interactions or enhancer promoter contacts or so on? And our thinking is that it seems like local accessibility can actually be explained, or chromatin accessibility can be pretty well explained using local DNA sequence alone. And most likely this gap in prediction is probably where we could gain information by looking at some of these other features of the gene regulation code. We can do a similar calculation to quantify performance on the profile task. And so for this, we use the Jensen-Shannon distance, which is basically a distance metric for probability distributions. In this case, lower is better. and we can get a distribution across all of our cell types. And so we use these types of metrics as well as some other kind of analyses to do a QC on these models. And after our QC, in this data set, we have basically trained a suite of Chrome BPNet models for 189 developmental cell types from human organs. And so I talked a little bit about how we can obtain these motif patterns, and I want to flesh that concept out a little bit more. And so traditionally, when we do experiments such as ChIP-seq or so on to assess transcription factor binding, we would represent binding preferences using a position weight matrix or a PWM. And the way these work is you get a set of enriched sequences. In this figure here, every row represents one sequence and every column is a position, so these sequences are aligned. And you can convert these observations of sequences to a position frequency matrix where we basically count each nucleotide or the number of the number of sequences with each nucleotide at each position. And then we can convert these to probabilities and scale them by the background frequencies in the genome of all of these nucleotides to obtain a position weight matrix. And these PWMs lend themselves to a nice representation when you subject them to an information content scaling where we can get basically a sequence logo where the height of the nucleotides represent their frequency among enriched sequences. So in our case, the model interpretation method lends itself to a different representation that we call contribution weight matrices or CWMs, and I'll show a bunch of these over the course of my talk. Like I mentioned before, the way we get these is by identifying these short loci with high contribution scores, like a short contiguous spans of high contributions. We align and cluster them. And then the way we can calculate the CWM is by basically taking the mean contribution score per nucleotide and position across these aligned and clustered genomic loci. And so that also lends itself to a nice logo representation where now the height of each nucleotide represents the average contribution of that nucleotide at that position to local chromatin accessibility across all the short loci that were basically clustered together to form that pattern. I'm just going to pause here and see if there's any questions before I move on. Okay, so now that we have this end-to-end kind of deep learning framework going from DNA sequence to predicted accessibility, and we can use that to extract the sequence rules that are driving accessibility, we can basically ask this question, well, what are the sequence determinants of chromatin accessibility during human development in these cell types? And what does that imply then about the transcription factors that bind these sequences? So when we clustered these motifs, these contribution weight matrices across the cell types in our fetal atlas, we obtain about 500 unique motifs across these cell types. And we've annotated and categorized these into different types of motifs. The canonical motif we're calling base motifs. These are motifs that basically represent or resemble known binding sites of transcription factors in databases. That represents about a fifth of our data set, our motif compendium. But we also have several hundred of these composite motifs, which are motif patterns constructed in exactly the same way, but which seem to have two or more binding sites for transcription factors. In some cases, those are two of the same transcription factors. We call these homocomposites. And in some cases, they're two different sites. We call these heterocomposites. And more than half of our de novo motif repertoire here are composite sites. And so we'll come back and kind of study these and try and understand what they imply about motif syntax. And then we also have a subset of motifs that don't seem to match known ones in our databases, and we're still trying to understand what these are. So I'm showing here the sequence logos for a subset of motifs. I like to show a few examples really large to be able to kind of appreciate how well the model-derived representations on the left, so these are the contribution weight matrices, often remarkably resemble the experimentally derived motif representations, the PWMs on the right from databases. And you can look through a few examples here to try and understand the similarities between the motif representations. And I'm just seeing a question here from Cesar. So the question is, would it be fair to say that these motifs are context specific, i.e. exhibiting a particular function adaptively, not just a generally characterized mechanism? So I think there's a couple different ways we can think about context. We could think about genomic context, so what types of regions in the genome do these motifs occur in? And then we could think about cell type context, so what cell types or organs do these occur in? And I will talk about that, both of those questions in the next couple of slides. So now that we have this motif repertoire set, we can basically ask what are some of the properties of these inferred transcription factor binding sites that are determining accessibility. We can ask about genomic context, we can ask about cell type context, and basically try and understand some of these global properties. So I'm going to show a lot of data on the slide, but I'm going to try and walk through it very slowly. In this figure, every row represents one category of these base motifs, which basically match known motifs. Every row is one motif. In the first column, we have the de novo neural network derived representation, the CWM. And then in the second column, we have a heat map representing the total number of genomic predictive instances across all cell types for each of these motifs. So like I said, we annotate these predictive regions in every peak in every cell type, and this is the total number of such predictive instances. Then we can ask what type of genomic regions do these motifs occur in, right? And so we could think about this in terms of whether motifs typically occur in more promoter regions, intronic, exonic, or distal regions, and those are represented by the bar plot here. So this is the proportion of motif instances for each of these motifs across different types of genomic features. And then a concordant metric would be what's the distribution of distances of these motif instances to the nearest transcription start site. And so that distribution of median distances is shown here. And then we could also ask about context in another way, right, in terms of what organs these motif instances are occurring in. And so this bar plot represents the proportion of motif instances that are detected in each of the 12 organs in our data set. The 12 organs in our data set, and you can see already some differences between very ubiquitous motifs and very cell context or organ context specific. And then finally, one really interesting property about these this neural network interpretation approach, or basically one interesting property about a neural network based strategy, and in particular this interpretation approach, is that we can pick up motifs that both positively, or features that both positively influence accessibility, but also negatively influence accessibility. I don't think I'll have time to get too much into the negative motifs here, but this last column is representing basically the class of motif, whether these features are typically associated with increased accessibility, which is the majority of the features, or this small subset of motifs that reduce accessibility. So I'm going to point out a few global features we notice. The first is that at the top of this figure, we have a number of motifs that predominantly occur in promoters. You can see they're dominated by the blue bar here. They tend to occur right up close to transcription start sites, and they tend to be ubiquitous across organs. And so again, to Cesar's question, in this case, these are a set of motifs that are very context-specific genomically, but not cell type context-specific. These are some of those motifs shown here. And what we find about these motifs is that they're promoter dominant, TSS proximal, but they also tend to be CG rich and include some examples of known methylation sensitive motifs. And one thing I thought was really exciting when I saw this is that the second motif here, BAN-P, was actually just recently characterized a couple of years ago by the Dirk Schubeler group. And so what's really interesting about this approach is we have a de novo approach for motif discovery that doesn't rely on what's known in the literature or databases. And so potentially some of these unresolved, unclear motifs we're picking up might represent similar uncharacterized factors. The second set of motifs, if we go down the list, tend to be much more distal. They mostly occur in intronic or distal regions outside of genes, and these include most of our tissue-specific motifs. They include factors like one-cut, FOX, the homeodomains, and so on that many might recognize from developmental biology or patterning or so on. And lastly, as I mentioned, we pick up this set of motifs that actually seem to negatively influence chromatin accessibility. In this case, we have 5 that seem to match known factors in terms of their binding sites, but they have this negative contribution to accessibility that we're still trying to understand. One really interesting feature of these is that when we look at total instances, these motifs have some of the most abundant predictive instances across the genome, so they're very frequent and they're often quite ubiquitous across many organs or cell types. Okay, so That describes basically how we can use our neural networks to first extract the rules that they've learned about sequences determining accessibility, cluster them to define the patterns or motifs, and then understand how these motif patterns have different contexts basically during development and also in the genome. And now I'm going to come back to the question I talked about at the beginning, which is how does this binding site syntax impact transcription factor synergy, at least at the level of chromatin accessibility? And so I'll spend the rest of my time on this point. So in order to talk about transcription factor synergy, let's discuss a little bit the different modes that could produce a synergy between transcription factor. And we could define synergy here as like a combined effect that deviates from independent effect of two factors. So one way to achieve transcription factor synergy would be through DNA-mediated mechanisms, for example, through direct interactions between two proteins. For example, when they form a protein complex, and thus they prefer to bind their sites at a very specific arrangement, which has favorable binding for the proteins in that complex. Another recently described phenomenon in the last few years is basically this case of DNA-mediated interactions, where you have two proteins or two or more proteins that form weak interactions in solution, but in fact, when their binding sites are arranged in a specific orientation on DNA, DNA actually acts as a partner in that interaction, stabilizing weak contacts between these transcription factors. So this would be another case where the binding site arrangement would impact synergy of transcription factors. We could also think about some modes of synergy that are nucleosome mediated, right? So nucleosome are these protein octamers where DNA is bound up around them. They can be stacked and block access to DNA. And so one way to get synergy would be through like a passive competition or a mass action model where transcription factors are basically competing with nucleosomes for access to DNA. And if one transcription factor is able to successfully compete, that will that will make the binding more favorable for a second transcription factor, and the readout would be some type of synergy. And then the last option here could be like an active remodeling, where a transcription factor recruits another factor, a chromatin remodeler, that actively evicts nucleosomes from DNA, kicks them off the DNA in order to facilitate binding for itself and other transcription factors. Each of these different modes of transcription factor synergy, these different mechanisms, has implications for DNA sequence and thus the syntax that we might find within it. So for DNA-mediated interactions, we might, in this case, expect some constraint on arrangement of motifs, i.e. syntax, and also we'd probably expect that this mode of synergy would probably interact at shorter distances within length scales of protein-protein interactions. Alternatively, in this nucleosome-mediated scenario, this type of competition or this type of synergy doesn't really depend on some specific binding site syntax. The more important feature here is, for example, how good is a transcription factor at competing with nucleosomes for DNA. And we'd also potentially expect this to act over longer distances. So among these, yeah, maybe I'll take Philippe's question first.
Hello, can you back up to the slide, please?
Yes.
So for any protein to stick on the DNA, on a DNA fragment, we first need this DNA region to be open. So don't we always first need the nucleosome to leave the DNA fragments?
It's a good question, and I think the answer is not exactly. So we could have this type of competition either through, so I think the second case is maybe consistent with what you said, where a transcription factor will recruit another factor that will evict the nucleosome, thus enabling it to bind its site, but still the nucleosome's gone before it can bind. So that's consistent. But we also think that DNA can sort of breathe around the nucleosome. It can unwrap and rewrap transiently. And in some cases, transcription factors might be better able to access or bind DNA, depending on specifically how it's wound around nucleosomes. And so in this case, there might still be a possibility of competition. So yes, the general rule is that transcription factors need accessible DNA to bind, but in practice, there are some more complexities, and this is an example.
Okay, so if we take the DNA mediated, it could be a situation where the nucleosome left somehow by itself because the DNA unwrapped.
Indeed, yeah, that's kind of how we're thinking about this case. I suppose you could also think of a more complex case where you have both DNA-mediated and nucleosome-mediated interactions, and I guess I'll actually show kind of some examples about that, but that's kind of how we're partitioning, yeah, these different modes of syntax or synergy.
Okay, thank you.
Thanks for the question. Okay, so we have these two different modes of synergy. I want to show one really, really cool example that was discovered by our colleagues in the Waisaka lab here at Stanford last year. So they basically serendipitously discovered this composite motif that they call coordinator, which has a homeodomain site and an e-box site. You can see there's kind of two motifs popping up in this PWM. And they found that this motif was enriched in mesenchymal tissue that gives rise to the face and the cranial in mesenchymal cell types that give rise to face and limb, basically. And they did a very beautiful study where they figured out exactly which factors bind these two sites. Transcription factor recognition of sequence is degenerate, so many families, large families might recognize a similar site. And so they figured out exactly which factors bind these two sequences. And then they actually solved an X-ray crystal structure and determined that this DNA with these binding sites in this particular arrangement with a fixed spacing between them actually facilitates interactions between an ALX4 protein and a twist1 TCF4 dimer, such that when DNA has their binding sites arranged at the specific syntax, you actually get a stronger contact between stabilizing this complex. And what was really neat is that they actually did experiments to show that the cooperative or synergistic binding kinetics of this complex depended on this fixation between motifs. So this is, I think, a really nice example where motif syntax was quite relevant for binding of specific complexes. And in this case, they have a very tissue or cell type specific role. So our question could be, given that such examples of transcription factor synergy and syntax, how could we systematically find such examples during development? And in particular, how could we use our machine learning models that have supposedly learned sequence rules of accessibility to help answer this question? So one of the things that these trained neural networks enable us to do is basically in silico experiments where we can push any new sequence through the models in a forward pass and obtain the output predicted accessibility. Because we can make predictions on new or synthetic sequences, we can basically use that as an experimental, as an in silico experimental platform. So the way that this works is that we would take a set of background sequences, like a library of non-accessible DNA that's matched for example, GC content with our peaks in a certain cell type. We can insert some sequence of interest, say a transcription factor motif, and then we push it in a forward pass through one trained cell type specific Chrome BPNet model. And because the model has two outputs, we will obtain both the set of profiles for this, say, 100 sequences, as well as the predicted counts. And we can basically take the predicted counts for the edited sequence, subtract the predicted counts for the background sequence in order to get an effect. And we can average this effect over these 100 sequences, over this library of sequences, essentially to marginalize out the effect of sequence context and isolate the effect of a specific sequence on accessibility. And so we can basically get these mean effects and mean predicted profiles for any given sequence. So this works, of course, for one sequence, but it's very flexible. You could add in whatever DNA sequence you want here, as long as it's within the same input length that's taken in by the Chrome BPNet models. So we could also imagine generalizing this approach to isolate the joint effects of two sequences on accessibility. And so instead of just inserting one motif, we could insert two motifs into our library of background non-accessible sequences at some k base pairs apart. In our case, we will loop over that value of k and vary it from 0 up to 200 base pairs. So basically, these motifs are being inserted in sequences at 0 and then 1 base pair apart and then 2 base pair apart and so on. And also, for any given set of motifs, they'll have some unique orientations relative to each other. So for two of the same motifs, there are 3 unique orientations. For 2 different motifs, there are 4 unique orientations. And so we can also exhaustively loop over every possible orientation of 2 motifs and predict accessibility. And so you might see where I'm going here, but basically we can systematically vary the positioning and spacing of motifs, 2 motifs relative to each other, look at the predicted effect on accessibility, and use that basically as a readout of whether these motifs, A, are having some type of synergy, at least at the level of accessibility, which is what we're measuring and predicting here, and B, does that synergy depend on some type of syntax? So here's an example of what one of those experiments would look like. In this case, we're looking at the same motif as the coordinator example that I showed you because it came up within our de novo motif discovery. So you have a homeodomain site and an e-box. The blue lines represent the positions of those motifs as we're moving them closer together within our genomic region, our synthetic genomic region. The dark gray represents the background accessibility with no motifs asserted. The red line that's moving represents the predicted accessibility for these 2 motifs together at each position or at each distance of the 2 motifs. And then the pink line represents the predicted accessibility for the optimal motif syntax. So this is an example of what we're calling like a hard syntax, where A, you have some synergy because you see that the effect of the 2 motifs is significantly greater than the independent effects. I'll show that quantification in a moment. But B, we also see that this synergy in this case actually depends on a very specific arrangement between these motifs. In this case, when they're 5 base pairs apart, and it's so specific that if you blink, you'll miss it. But in other words, when these motifs are at some specific arrangement, we see this spike in predicted accessibility. And we can quantify those basically in order to define whether motifs fall into this case of synergy or syntax. And so to define synergy, we basically look at the distribution of the effects of those 2 motifs across all of our 100 background sequences, that's shown in red here, and compare that to the distribution of effects for the 2 motifs, each motif alone, so that's shown in these right 2 box plots, as well as the sum of the independent effects. And so we define a case of synergy when the joint effect is significantly greater than the sum of the independent effects. And then secondly, we can show a quantification of kind of the movie I showed on the previous slide, where in this plot, every bar represents one set of distance between 2 motifs. Every plot is one orientation of the 2 motifs, and the height of the bar represents the joint effect of those 2 motifs. And so what we want to identify is these cases where you see a spike basically in the joint effects, or in other words, we can define that as we can define these cases of syntax as cases where some specific arrangement of the 2 motifs of the joint effects differ significantly from the mean. So we basically do a z-score across of these joint effects across all positions and orientations and ask whether we have a high z-score. And so this approach basically allows us to do a systematic analysis where we can quantitatively define both synergy, a case of predicted synergy, as well as when that synergy is actually syntax dependent. Like I said, the motif I showed in this example actually matches, resembles the coordinator motif. And what was really remarkable for us to see is that our prediction of synergy in this case with a preferred 5 base pair spacing actually exactly matched the very restricted binding site arrangement for the coordinator motif, where that binding site arrangement was experimentally derived and then also validated. And like I said, in this case, the cooperativity between these 2 transcription factors was actually fully dependent on this strict spacing and arrangement between motifs. So yeah, this is one example of basically what a type of like hard syntax would look like, where you have both synergy, but also it depends or is mediated by a specific arrangement or syntax of binding sites. We also see cases like this, like a soft syntax, where you have this synergy. And so this is a similar type of animation as I showed before. As these motifs are being moved closer together, they have an increasing joint effect until some certain point. But in this case, it's not really restricted to some specific motif arrangement, right? As these motifs are moved closer together, they gradually show a greater joint effect. And so in this case, we call this an example of like soft syntax, where you have synergy, but it's independent of some specific binding site arrangement. And in this case, whereas in the previous example of hard syntax, we would expect that to be more consistent with a model of DNA-mediated synergy, this could be more consistent with a model of nucleosome-mediated synergy. So this approach basically allows us to do an in silico evaluation of motif cooperativity and syntax based on basically using all of the composite motifs that we discovered in our de novo motif discovery. And so we tested around 130 motifs total. In this plot on the left, every point represents one motif, and it's kind of has a shape according to its classification in our analysis. And it's plotted in a scatter plot with its joint effect versus its independent effect. And so you can see that in the very strong cases, these motifs with synergy have a significantly greater joint effect compared to their independent effects. Generally what we find is that as these motifs are moved further apart, or as the syntax is broken, the joint effects essentially decay to the sum of independent effects. And so in these plots, the red lines represent the predicted joint effect, the total counts basically at each distance between motifs going along the x-axis. The gray lines represent the sum of independent effects. And you have a couple examples of hard syntax, soft syntax, and no synergy. What you see in the case of hard syntax is a specific spike at a very particular distance between motifs. And then at all other cases, we essentially have a joint effect that matches the independent effects. In the case of soft syntax, as motifs are moved further apart, the joint effect kind of decays to the independent effects. And in this case of no synergy, the motifs don't really care that they're near each other. So I'm just going to go back to the questions here. So what is a soft effect? Yeah, so basically the way we define a hard and soft effect is when our hard syntax is when you have a joint effect at some arrangements that are significantly greater than the mean of arrangements. And I think these plots kind of answer the question of Philippe, where it's not always simply that the motifs joint effect decays as they move apart. In some cases, it really is a very specific joint effect at some arrangement of motifs. And this is kind of one example here where the motifs don't care if they're necessarily close to each other. They care that they're in some specific arrangement. And so that's how we would define a hard syntax. In the other hand, this soft syntax is where you have a sort of more of a decay of predicted joint effects as these motifs are moved further and further apart. So hopefully that answers the question there.
Well, sorry.
Yeah.
It's still, of course. Well, I understand the difference between hard and soft, but you also have a shape for hard and soft. The third category on the top left plot, like this
Indeed.
diamond.
Yeah, we have exactly. So in this case, yes, in this case, we have a few motifs that fall into this category where they have both a hard and soft. So I don't have an example showing that here, but in those cases, yes, those would fall into the case you described where there's some spike in predicted accessibility in the joint effect of these motifs when they're at some specific arrangement. However, we also see the same effect that there is some decay as motifs move further and further apart. And so because of these quantifications, we could kind of categorize precisely these maybe like 4 different types of behaviors. But yeah, that's a good point.
Okay, thank you.
Yeah, yep. Okay, so then the last question I'll talk about here is I sort of alluded a lot of the question of how cell type or context specific are these synergistic or interactions. In the previous analysis, what we did is we took for every composite motif, we analyzed its synergy using this in silico framework through one model for one cell type, basically the one where it had the most predictive instances. But in this case, we can actually ask, are these observed cooperativity or synergy effects cell type specific? And this would get us to an analysis that basically could identify these cases similar to the coordinator motif, where perhaps you have some synergistic syntax-mediated interaction that's very important for development of some specific gene program. So this is another kind of variant on our same in silico marginalization strategy, where we will, again, take background sequences, insert motifs at different distances, except now, instead of pushing them through just one ChromeVPNet model, we actually do push them through all 189 of our suite of trained ChromeVPNet models. And so what you get as an output is basically 189 effects, one per cell type. And so I'll show a couple examples of what that looks like. In this left-hand plot, we're looking at the predicted accessibility profile as a heat map over 1,000 base pairs. So every row is one cell type, and I've just plotted the top 40 or 30 or 40 or so cell types with the highest predicted effect. The second plot represents the effect as a fold change from background sequences, the joint effect of those two motifs. And then lastly, because we actually have a multimodal data set where we have gene expression levels, we can look at expression levels of transcription factors that could possibly bind these motifs. So this is an example of an IKZF1 runx motif. We looked at the IKZF factors as well as the runx. And generally what we find in this case is that in the cell types where you have the highest predicted effect of these two motifs, we also have generally the highest expression of transcription factors that could bind those motifs. In this case, we don't really see any cell type specificity, any specificity of which transcription factor it could be. And so it's possible that all of these factors are expressed in these cell types, mostly immune cell types from the thymus and spleen in this case, and they could all participate possibly in this synergy. But we also have another example where we can use these expression levels to actually nominate candidate transcription factors that could bind these motifs in certain cell type specific cases. So this is a very similar figure where on the left you have the predicted accessibility as a heat map in each of these cell types for the top 30 or so. We have the predicted accessibility in terms of the predicted counts or the effect on counts. And then finally, we have expression of all the members of the Fox family that we detected, as well as the HNF4 motifs that could possibly bind this Fox HNF4 composite site. And in this case, we had a couple Fox transcription factors that were actually fairly specifically expressed in these cell types. And so it's possible here that we could use expression to kind of nominate these factors as the ones that might be participating in this synergistic interaction. And so we think that this in silico framework provides a nice way to screen essentially millions of sequences in minutes or hours that would allow us to compare effects per cell type and ultimately nominate some more detailed hypotheses that could then be validated in the appropriate cell systems or cell culture systems. And in this case, we think that the cell type specific cooperativity where you only see a predicted joint effect in some cell types means that these models have only learned that these motifs are relevant in certain cell types, in this case in the liver, and likely can be explained parsimoniously by cell type specific expression of those factors. So I think I'll wrap up there. Basically, what I've talked about today is how we can use a deep learning framework to identify transcription factor binding sites which influence chromatin accessibility. And we've applied this in an atlas of human field development to understand specifically which are the relevant sites during development. We can use these models together with model interpretation techniques to run a de novo motif discovery, which basically picks up both known and novel motifs, including some that might reduce accessibility. And lastly, we can use these types of trained suites of models as a platform for in silico experiments, in this case, to nominate cases of synergy and syntax. And this was a huge project and I couldn't talk about many parts of it, but in some of the other work on the same project, we worked on describing the regulatory landscape across these cell types and across the genome. We show how the enhancers that come up in this analysis can often be validated in vivo. And we also can use these same models to predict variant effects. And yeah, I'll just say really quickly, I think this is like a really cool rich field where these sequence to function types of models are gaining a lot of traction, being used in lots of tasks. Maybe there's a way for you to use them in your research. Some of the major applications are basically interpretation of sequence rules and syntax, which is what I talked about today. We can also do these types of counterfactual and in silico experiments, asking what if we saw something else in the DNA. One really cool application this is to predict the effects of non-coding variants on accessibility, which are notoriously hard to interpret. And then a final application is basically designing new DNA sequences with some desired readout, like cell type specific accessibility or activity. And so these are some of the cool work being done in this area. And lastly, I just want to thank my advisors, Will and Anshul, and my co-first authors, Betty, Sam, and Yanting, and our funding sources, and to QLS for having me back. And I'll be happy to answer any more questions. Thanks.
Thank you, Selin. So if we have any questions, please, Philip.
I was uploading, but
Oh, sorry.
but
Sorry, I thought that was a raised hand.
I do have questions. I have two questions. The first one is is pretty simple. Can you go back to slide 19?
Yeah.
Please. It's just a question of how do you align exactly the sequence on the top left? Because like, do you align the sequence on like a base sequence like genome 1839, or do you align them between them?
Correct. It's the latter. It's like a more abstract space of relative alignment, not a genomic alignment.
Okay, so
Yeah.
but so you have to decide what because you have to decide what site you're targeting and then align them on that specific site, right?
Exactly. So there's actually two. I wonder if I have another schematic here that would explain this. No, I don't think I do. Okay, so let me just go to a different slide here. There's actually two separate steps here. There's one step which involves basically identifying these spans of high contribution, chopping them out, clustering and aligning them. That technique is called that algorithm is called TF-Medisco, described in this paper. And in that case, the output is just the clustered patterns where the sequences are aligned relative to each other. Then to go back to the genome, we basically use a second algorithm called Finemo, also developed in the Kundaji lab, which basically takes these matrices and runs like a sparse linear model along the contribution scores, trying to explain the observed contribution scores as combinations of the motif patterns. So there's two separate algorithms, one to define the motif patterns and a second to map them back to the genomic sites. That makes sense?
Yeah. Thank you. And last question. So you started by showing this convolutional neural net. Have you challenged it with new models and maybe with some new trendy LLMs?
Transformers, indeed. Yeah, that's a good question too. Yeah, so I haven't worked on the benchmarking of this specifically, but I'm happy to point you to some papers where it seems like for the local accessibility prediction task, Chrome BPNet performs just as well as some larger transformer-based models. Our paradigm in the lab, and I think this is really like Anshul's strategy, is that often the simplest model that can achieve the task is the best one for the job and usually the most interpretable. So that's why we've stuck with this architecture, but it's a great question. And yeah, there's active work in other parts of the lab trying to explore where the gains you could get just simply from better architectures.
Thank you.
Yeah. And to Cesar's question, I forgot to say that we are hoping to submit this work within the next week or so. And so, yeah, I'm happy to share the slides, but also keep an eye out for a preprint, and that should have all the references as well.
And please note the seminar is being recorded.
Oh, yeah, there you go.
Yeah.
Perfect. Great. Any other questions?
Seeing no other questions, Selin, thanks so much for a wonderful presentation. We're very happy to have you back, and we hope to have you back in person soon.
Sounds good.
Cool.
Awesome.
Thank you, everyone. That's the conclusion of the seminar series for 24-25. So thank you for finishing it on a very high note. And everybody is encouraged to come to the 3MT this afternoon at 4 o'clock in the QLS conference room.
Awesome. Have a great summer, everyone. And thanks so much for having me.
So, did you see everybody waving to you from
I
the?
did. I
Yeah.
saw the LDI satellite
Exactly.
satellite session.
Cool.
That's awesome.
Thanks.
Okay, bye everyone. Thank you.
Bye.